Hello, everyone. Thank you for joining today's webinar, Redefining NASH Drug Discovery, a New Approach for Complex Disease Modeling. My name is Sue Grepper. I'm a senior application scientist for InSphero, located on the west coast of the US. And I imagine, like you, we are very interested in continuing the discussion around NASH. As you've seen in the news, people that have NASH and other related cardiometabolic disorders are more susceptible to other types of injuries, such as viral infection. And so there's an even greater need now to try to find a cure for people suffering from NASH and these cardiometabolic diseases. So thank you for joining. You may see that you can, as attendees, ask questions at any point, and we would love to hear your questions and comments as we go through the webinar. My colleagues will be kind of compiling the questions as we go, and I hope you'll stick around at the end and we'll go through some of these questions. So thank you again, and let's get started. So I'd like to start with a few definitions to help set the stage. Non-alcoholic fatty liver disease, or NAFLD, is the hepatic manifestation of the metabolic syndrome and is very closely associated with type 2 diabetes, insulin resistance, and obesity. NAFLD is characterized by the development of simple steatosis, otherwise known as non-alcoholic fatty liver. The steatotic hallmarks of fatty liver include bloated hepatocytes, displaced nuclei, and fat accumulation. It is estimated that 25% of the worldwide adult population currently has fatty liver, equating to almost 1 billion people. While that statistic may sound shocking, it should be pointed out that fatty liver by itself is benign and can remain dormant for decades. However, in approximately 30% of these patients, which then equates to over 250 million people, the disease may progress to include inflammation and degeneration of hepatocytes. This more advanced stage is referred to as non-alcoholic steatohepatitis, or NASH. NASH-related hepatic injury and inflammation can lead to the development of fibrosis, which is the deposition and unclearing of collagen fiber scar tissue through the activation of the liver stellate cells. Once the scarring is extensive enough that it blocks proper blood flow in the liver, these patients are considered to have cirrhosis, and their only solution is liver transplant. So I'd like to add a few important facts here. NASH patients are now the most frequently requested recipients for liver transplants, beyond that of hepatitis B and C. In addition to potential cirrhosis, these patients are at much greater risk for developing hepatocellular carcinoma. And as I mentioned earlier, because of their compromised state, these patients are also at much greater risk for serious complications stemming from viral infections. So as you can see, NASH is something to be taken very seriously. Unfortunately, there are no NASH therapies yet available on the market, and there is a need for predictive preclinical models that recapitulate the complex pathways of NASH progression. As you can imagine, this is no easy task, and it can take decades to develop in humans. There are currently numerous in vivo models used for preclinical development. These include diet-based, chemically treated, and/or genetically altered animal models, typically mice. Many of these can be useful for incorporating extrahepatic systemic pathways related to NASH, such as diabetes and obesity. However, these studies can take several months and are very expensive. In some cases, these animal models don't fully reflect the regulation of human pathways or how humans would respond to therapeutics. Scientifically speaking, an ideal preclinical model should recapitulate the pathophysiology of human disease. And from a cost and time perspective, the model should enable rapid screening of large numbers of compounds, as well as combinatorial treatments. That leads me to introduce the primary human-based platforms we've been developing over the past several years in InSphero. As you may already know, we had initially developed a primary human liver model for toxicology testing over a decade now, using our patented hanging drop technology. This technology allows cells to self-aggregate in a scaffold-free environment, and we found it was perfect for liver cells that would otherwise not culture well in 2D. For example, in my previous job, when I was doing inflammation-related toxicology research, I had the arduous task of trying to get primary human hepatocytes and primary Kupffer cells to co-culture in a dish. It was really difficult because hepatocytes require a collagen basement membrane in order to attach, where Kupffer cells didn't. Hepatocytes did a much better job surviving in Williams E medium, where Kupffer cells preferred a macrophage medium such as RPMI. Once I moved to InSphero, I was very pleasantly surprised to see that allowing the cells to self-aggregate and a proprietary media allowed them to naturally form tight cellular connections that are critical for cell longevity and function. So, building upon this initial model, we took on the task of developing a much more complex model to study NASH. This journey has taken us almost 4 years. First, it was critical to include primary stellate cells in addition to the hepatocytes and epithelial cells from the tox model. Once we had succeeded in finding the right combination of cells and were able to maintain them in an unactivated state, we needed to recreate the NASH phenotype in these microtissues. We tried various stimuli, such as saturated and unsaturated free fatty acids seen in clinical NASH patients to mimic the steatosis-like phenotype. We optimized inflammatory stimuli that represents the leaky gut inflammation associated with NASH. And lastly, we optimized the media to contain diabetic levels for different sugars. This complex combination of stimuli was necessary for development of not only the steatotic and inflammation aspects of NASH, but also the downstream fibrosis phenotype. Of course, in order to determine whether we'd achieve these different hallmarks of NASH, we needed to find appropriate endpoints that could be measured from our small microtissues, which contain less than 5,000 cells. Not all endpoints are feasible, such as Western blots, which would likely require an entire plate of microtissues per treatment group. However, there are numerous endpoints that can be measured. For example, histology, high content analysis, gene expression, which with the most recent advanced technologies, only a single microtissue is required per treatment group for RNAseq. These endpoints are not considered high throughput, however. For rapid endpoint assessment of numerous compounds and combinations of compounds, we ultimately wanted to find predictive and screenable biochemical assays for the different hallmarks of NASH. These include intracellular triglyceride expression, or TG, the use of Luminex panels for cytokine expression, and cleaved C-terminal procollagen 1 secretion. With these assays, we can see clear differences between healthy and lean media treatment groups to that of the NASH stimuli treatment groups. Of course, the ultimate goal of developing this model was to allow for testing potential therapeutics for their ability to block different NASH pathways within the liver. In the next slides, I'll describe the process we used to optimize this NASH model so that there were enough differences between the healthy and diseased microtissues. This was critical because we need that window to determine drug efficacy. So this is what we found to be optimal for this model. For determining compound efficacy against NASH disease progression, the drug or vehicle control is added at the same time as the NASH stimuli induction, meaning the lipid loading, inflammatory stimuli, and high sugars over the course of 10 days. The control-treated tissues are cultured in medium with normal lean glucose levels, mimicking the condition in healthy people. Hence, I'll refer to this treatment group as the lean group. We have found that measuring certain parameters at day 5 versus day 10 give the most useful information from our model. For example, we found that at day 5, the release of inflammatory markers due to NASH stimuli is at its peak, whereas we see the greatest window between lean and NASH treatment groups for fibrosis and steatosis-related endpoints at day 10. So, keeping in mind those optimized cell combinations, stimuli, and endpoint timing, we wanted to know whether we can induce these 3 phenotypes of NASH in our microtissues. First, we wanted to see whether we could induce lipid accumulation within the tissues treated with the NASH stimuli. Using whole mount HCI, we performed Nile red staining of the lipids within the cells after 10 days of treatment with the lean control versus NASH conditions. What you see here are 2 microtissues treated with lean media conditions on the left column and 2 microtissues treated with NASH stimuli conditions in the right column. The nuclei are stained blue DAPI stain and the lipids are stained with Nile red. Nile red or NASH treatment clearly increased the lipid accumulation within the hepatocytes, and the steatosis-like phenotype was observed. Then we also measured the triglyceride levels within the tissues in the lean control and NASH conditions. In the NASH conditions, we observed around 2-fold increase in triglyceride levels as compared to lean control. The triglyceride results are really exciting to us because, frankly, these images are exciting to look at and make for pretty pictures and publications, but it's quite labor intensive, especially if you intend to quantify the differences. So having a biochemical measurement is quantitative and much quicker. As I mentioned earlier, the second major hallmark in NASH is inflammation. And so an important part of our optimization was to show that our NASH model will reflect inflammation as has been shown in the clinic. The release of different pro-inflammatory cytokines and chemokines were measured at day 5 of treatment using Luminex multiplex technology. Secretion of cytokines IL-6, TNF-alpha, and IP-10 were increased in the NASH condition as compared to the lean control. Similarly, the secretion of chemokines IL-8, MCP-1, and MIP-1A were increased in the NASH conditions as compared to control. As a quick review, chemokines are specialized cytokines that call out to blood macrophages for them to infiltrate the liver upon damage. These same cytokines and chemokines were found to be elevated in the plasma of patients with NASH. I should point out here that some of the cytokines and chemokine values in the control treated groups were below the detection limit, which means that the Cooper cells were appropriately unactivated in the lean conditions, and that is why we could not perform any statistical analyses between the treatment groups. Lastly, we wanted to optimize the fibrotic aspect of NASH within our model. As you can imagine, it was the most difficult to obtain within a 10-day window, as it takes humans decades to reach a stage of compromised liver. After testing numerous stimuli conditions and numerous kits, we finally found a kit that nicely measures the secretion of cleaved C-terminal propeptide from collagen type 1 as a marker of fibrosis. As you can see from this graph, NASH stimuli treatment significantly increases the secretion of pro-collagen type 1 by over 10-fold. So those were the 3 major hallmarks we felt were critical for our model. In addition, we felt that we could gain a ton of knowledge from RNA from lean or NASH treatment groups. Therefore, we performed gene expression analyses using targeted RNAseq technology, covering the whole transcriptome. As I mentioned earlier, this method can be performed with single microtissues. RNAseq now allows the generation of large data sets and many replications from a small amount of biological sample. What you see here is a principal component analysis, otherwise known as PCA, based on whole transcriptome expression profile. Each dot represents 1 sample, respectively 1 microtissue, and 10 microtissues for each condition. For those not familiar with PCA plots, these plots display differences between samples. In other words, the closer the dots are, the more similar they are regarding gene expression. In this plot, we clearly see distinct clustering of the NASH versus lean samples at both day 5 and day 10, indicating significant changes in gene expression profiles between the 2 treatment groups. You can see some overlap between lean day 5 and lean day 10. This is expected and makes perfect sense. Interestingly, 5 and 10-day NASH samples are quite separated, which might represent the progression of NASH over the course of the 10-day period. Based on that large generated data set, we also performed gene expression analyses. As an example, this figure shows the top 20 most significantly regulated pathways in NASH versus lean at day 10 of treatment. Red bars representing the top 20 most upregulated pathways, and blue bars representing the top 20 most downregulated pathways. Several of these significantly regulated pathways are associated with inflammatory and metabolic pathways, as highlighted in the far right. Some of these pathways most downregulated after NASH treatment include PPAR, signaling pathways, fatty acid metabolism, and cholesterol biosynthesis. We feel that this data set confirms the induction of a NASH phenotype on transcriptomic level and shows a clear regulation of metabolic and inflammatory pathways. To further confirm the NASH phenotype, we analyzed gene expression changes within key pathways involved in NASH pathogenesis. Here you can see PCA plots generated for lean and NASH samples at day 10. Again, 1 dot equals 1 microtissue and 10 microtissues tested per treatment group. Each PCA plot is depicting a defined pathway, fatty acid metabolism, inflammatory response, or TGF-beta signaling. The principal components were calculated for only genes involved in a pathway of interest. As you can see, these data show that the tissues from the lean control treatment group in blue dots were clustering together, as were the NASH treatment group in green dots. Furthermore, the 2 treatment groups were separated from each other in respect to the specific pathways analyzed. Again, we feel that these data support the NASH phenotype at the gene expression level. Now moving on to therapeutics and development for NASH. I wanted to highlight this great cartoon by Monica Connerman at the University of Oxford, as it nicely illustrates the numerous pathways researchers are focusing on for the treatment of NASH. As you can see, the liver is the obvious main organ highlighted in the center of this illustration. We've successfully tested several drugs representing these liver-based pathways in our NASH model, including ACC inhibitors such as firsocostat, PPAR alpha delta agonists such as elafibranor, and ASK1 inhibitors such as selonsertib. I will point out, however, there are other organs shown here that also play a role in NASH initiation and/or progression, and we recognize that these pathways are not represented in our current NASH model. For example, FXR agonists, such as Intercept's OCA, are among some of the hottest NASH drugs in clinical trials right now. Some FXR agonists have some direct effects on the liver, but the main site of action of most FXR agonists are in the intestine, causing an increased release of FGF19, which then binds to FGFR4 in the liver, inhibiting CYP71, and ultimately decreasing the conversion of cholesterol to bile acids. So, without the intestinal FGF19 input, we're only able to measure partial effects of this particular class of therapeutics. Of course, in this circumstance, we could add in FGF19 alongside the FXR agonist, which is something we're looking into. Cinacriviroc is another type of therapy that we've tested, and in this case, didn't expect to see much of an effect. Allergan cinacriviroc is a CCR2, CCR5 inhibitor, which works by blocking the signal of the blood macrophages to infiltrate the liver. Our model does not currently have blood macrophages, so we didn't expect to see a significant effect beyond the small cytokine effect from binding to stellate cells and Kupffer cell, CCR2, CCR5. Long story short, because NASH is such a complicated disease, it's important to choose the right preclinical models for testing potential therapeutics. I thought this schematic might also be interesting to show. This schematic was pulled together from data from clinicaltrials.com and assembled by the IQVIA Consulting Institute. As compared to the previous cartoon, this one grouped the therapeutics based on metabolic pathways in blue dots, inflammatory pathways in green dots, or fibrotic pathways in yellow dots. Then also what stage clinical trial these therapeutics are currently in. It may be difficult to see here, but all the therapeutics on the outer ring are in phase 1 trials, and then moving inward, the therapeutics on the inner ring are in phase 3 trials. These data were assembled in 2019, so there are certainly additions that could be added to this list, and some trials have subsequently failed in the sense of missing primary endpoints. However, I thought it is interesting to show how most compounds are targeting only one of these 3 major hallmarks of NASH, and now we're starting to see combination trials which combine drugs from different pathways in order to have the greatest effect for these patients. And to reiterate what I mentioned at the beginning of this webinar, scientists believe that people can live with fatty liver and some inflammation. It's the downstream fibrosis that will need to be blocked or resolved in order to prevent possible liver transplantation and/or hepatocellular carcinoma. Since I wanted to make sure there was time for questions afterwards, I'm now going to focus on the fibrotic aspect of our current NASH model. In order to demonstrate blocking the progression of fibrosis in our model, we have extensively studied a tool compound which is an activin receptor kinase, or ALK5, inhibitor. This inhibitor blocks the binding of TGF-beta to its receptor, blocking the certain downstream phosphorylation events, which ultimately lead to stellate cell activation, collagen deposition, and fibrosis. To demonstrate the effect of the ALK5 inhibitor in our model, we first visualized this at a histological level using serous red staining. As you can imagine, performing IHC and serous red staining is not high throughput. It requires pooling of 20 to 30 microtissues per treatment group and is difficult and time-consuming to quantify. As you can see in the images shown, we combine numerous microtissues after treatment with either lean control media, NASH stimuli, or NASH stimuli with concurrent ALK5 inhibitor treatment. We found that it's difficult to assess differences between bright field images unless there's a striking effect of a drug, which there is here. Instead, we found that if you take an inverse image of the polarized light images, it will allow visualization of the collagen fiber deposition that is associated with NASH-related fibrosis. Although we've not done this for this particular data set, these images can be quantified using software such as Cell Profiler or ImageJ. There are additional advanced softwares we're looking into that can not only quantify serous red, but also IHC of collagens and other markers of fibrosis. For the sake of this presentation, I hope you will agree that there's a noticeable difference in the increased collagen fiber deposition after NASH treatment, and this is blocked by concurrent treatment with ALK5 inhibitor. However, and you'll notice this is a recurring theme of mine, Pro-collagen secretion is much quicker and much easier to quantify. As you can see from this graph on the right, NASH stimuli treatment significantly increases the secretion of pro-collagen type 1 by over 10-fold that of lean control. Concurrent treatment with the ALK5 inhibitor decreased the NASH-induced secretion of pro-collagen type 1 in a significant and dose-dependent manner. Again, since we have access to RNA-seq technology, we were able to analyze the effects of the ALK5 inhibitor on gene pathway expression in our NASH model. Using RNA-seq as previously described, we determined the top 20 most significantly upregulated pathways, the red bars, and top 20 most significantly downregulated pathways, blue bars. We pulled out a few of these pathways on the right-hand side. We can see that the ALK5 inhibitor treatment caused a clear downregulation of TGF-beta and integrin pathways, extracellular matrix deposition pathways, and in general, pro-fibrotic pathways, confirming the anti-fibrotic activity of this tool compound. On a more detailed level, we went beyond pathways and looked at the gene changes after ALK5 inhibitor treatment that were greater than twofold. You can see here NASH samples in red dots and ALK5 inhibitor in green dots. Among the top 20 most significantly regulated genes, there were several related to fibrosis. You can see here gene expression differences in different collagens, such as collagen 1A1, 1A2, 3A1. We also see differences with MFAP2, MFAP5, as well as TIMP3. These data reveal a strong regulation of fibrosis-related genes after ALK5 inhibitor treatment. While we have limited time for this presentation, I was still hoping to highlight and summarize effects that we've seen from some clinical therapeutics we've tested in our model that represent different target pathways. The compounds listed here are grouped here because they now serve as positive controls for the 3 NASH hallmarks as we've discussed in our ongoing studies. For example, firsocostat, an ACC inhibitor, inhibits a crucial step in the de novo lipogenesis pathway, and it rightly showed a strong expected effect on triglyceride levels. It also showed an effect on procollagen secretion. Because of this, we are currently recommending firsocostat as the positive control for steatosis in our services that we perform. Elafibranor is a dual PPAR alpha delta agonist currently in phase 3 clinical trials and may be the first NASH drug approved on the market. If I recall correctly, the primary endpoint of the current RESOLVE-IT clinical trial is resolution of NASH without worsening of fibrosis. This makes sense as a strategy because PPAR alpha agonists address liver inflammation, insulin sensitivity, but do not have direct effects known on fibrosis. In our NASH model, elafibranor did not show significant increases in triglycerides or procollagen secretion, but it did significantly affect cytokine and chemokine expression, and therefore serves as a positive control for inflammation. And I've already showed some effects of the ALK5 inhibitor in our NASH model, and is our current positive control for fibrosis. An interesting note is that while we expected the ALK5 to inhibit procollagen secretion, as we've reproduced this numerous times, we were also not surprised to see that this compound causes an increase in cytokines and chemokines. We think this is because TGF-beta itself causes a decrease in cytokine and chemokine secretion in the liver. So in this situation, we are inhibiting the inhibitor and therefore see an indirect increase in cytokines, chemokines after ALK5 inhibitor treatment. For many of the compounds we've tested in our model, we also performed RNA-seq-based gene expression analyses to get a more detailed insight into the compound's effects. This is a very large data set that could take an entire dedicated webinar, but I'd still like to give it a brief overview. You can see here a PCA plot based on whole transcriptomic changes for lean samples, orange dots, NASH samples in pink dots, and NASH plus 7 different compound treated samples in different colors. The PCA plot shows a clear clustering of each treatment group. This is very important to us because it indicates significant effects of compounds on gene expression patterns, but also different mechanisms of actions, since we observe a distinct cluster for each compound. I should point out that, except for the ALK5 inhibitor tool compound, these compounds are all in clinical development. Together with the data from our biochemical assays that I'd be happy to share offline when we have more time for discussion, these results provide detailed insight into compound efficacy on various parameters of NASH. They also allow details into the mechanism of action and a genetic signature in a human relevant in vitro model. So, in conclusion, I hope I've shared some interesting information around NASH drug discovery modeling in 3 dimensions. We feel that the 3D NASH model we have developed over the past several years recapitulates the three major aspects of NASH pathogenesis. This includes robust biochemical endpoints as well as whole transcriptome analysis. Not only were we able to confirm induction of the NASH phenotype, but now we can also offer scalable assessment of compounds' effects on this NASH phenotype. As I mentioned earlier, our current NASH model is not appropriate for studying systemic pathways associated with NASH. However, we have performed numerous proof of concept studies with NASH or liver targeting antifibrotic, anti-steatotic, and anti-inflammatory compounds. In summary, our human liver NASH model does enable researchers to quickly and efficiently screen NASH potential therapeutics, as well as perform combinatorial drug treatments. This last point is particularly important as the subpar clinical data reading out from monotherapy agents are suggesting that combo therapy might be necessary for treating NASH. The amount of time and cost to run combination studies in vivo animal models could be quite expensive and extensive. So that is where our model could be especially useful. But of course, the intent of this presentation was not meant to be a sales pitch for our model, but rather to continue the discussion of different models and endpoints for understanding the serious disease. So I hope you've been submitting some questions that I and my colleagues joining in can now address. Of course, if you would prefer to continue this discussion offline, please do note my email here. I'd also like to add that we will be sending you a follow-up email with links to additional information if you have interest. But I hope you'll stick around for a few more minutes so we can address any questions that you have posted. Thank you again for listening.